currently this is a pill. Currently this is a daily pill.
And can you talk about Nivalis just?
So Nivalis is the company that is developing this. So they are one of the drug companies that's working closely with the TDN, and this is their candidate drug. So they are funding the trial, and eventually they hold the patent on this drug, and they eventually hope that if it works, that they'll be able to sell it. Okay, so that's one arena. Other ways that we're looking at improving things going forward. VX661 is you might want to think about it as sort of the next generation of Lumacaftor. It is another drug that works very similarly. It helps folding in the cell, as we saw. It has fewer drug-drug interactions. It has a longer half-life. And there is phase 2 data, that's already been studied, where it did seem to help patients with F508del. So these were actually mostly homozygotes. Don't worry about the heterozygotes with the gating mutations, but there was this is FEV change in FEV1% predicted, and you'll notice, at higher doses, we're not just talking about 3-4%. We're talking maybe 7%, 9%. This may be a slightly better drug from an FEV1 point of view. We don't know yet, but it certainly doesn't seem to hurt, and it may have some benefits to. It may have some benefits to making to dosing and fewer drug interactions, et cetera, et cetera, et cetera. And there's actually some interesting data, test tube data, once again, about the potential that Lumacaftor and Ivacaftor, although they both seem that we think of them as helping each other, they may not entirely help each other, and there's a possibility that they're actually having negative effects on each other, and that VX661 in place of Lumacaftor may work better because it just doesn't. There's not any interference between these two drugs. So there are actually multiple phase 3 trials going on of VX661 and Ivacaftor, so the next generation of Orkambi, that are in various combinations of patients with, but homozygotes, patients who are heterozygotes who have specific types of mutations, and patients who have heterozygotes without these other specific types of mutation. These are all phase 3 trials. We expect data from them in the next 6 months to a year. It is quite possible that we'll have a second generation of Orkambi coming out in the next year that may work better. Really exciting. We don't know yet. Stay tuned.
Would the second generation of Orkambi replace the first generation?
Well, that's another big question. I think the real question is going to be how do these two compare in actual real world use? Are there other side effects that we find out that come in? It'll give us another option. And so that there'll be the possibility of having Orkambi and a different drug. And if you didn't do well with Orkambi, maybe you'll do better with another drug. We don't know the answer to that, and I would think up front the likelihood is that they'll both be available, but who knows? Is this going to replace Orkambi? We don't have the data yet. Other questions?
I have another question.
Yep.
At what point, if any, is the sweat chloride retested after starting Orkambi?
So we don't act. Oh yes, repeat the question. So the question was, at what point do we retest sweat chloride after starting Orkambi? And actually, in clinical practice, we do not. Sweat chloride measurement is really a research tool. It proves the diagnosis of CF when you have a child who does this baby have CF or not. Doing a sweat chloride will help you. In terms of actually using it in clinical practice to make decisions about how we treat patients, that is not done. And so everyone who gets started on Orkambi, either they take the Orkambi and they do well with it, or they take the Orkambi and they have side effects and they have to stop it. But we don't check their sweat chloride after they start it in general. Okay.
One quick one. Do you know if either of the two tracks you've just discussed are similarly being co-funded by the CF Foundation and these drug companies with sort of rights the same way? Sort of, the CF Foundation was brilliant in terms of sort of selling future rights to Orkambi and Kalydeco and making over $1 billion.
Yep.
I'm curious about this one and others. Is it a similar model or not?
Well, and it depends a lot on the specifics of the specifics, and I don't know. I can't answer. In general, the CF Foundation has done a fantastic job of partnering with industry. But in terms of who exactly holds what percentage of the rights to each of these compounds, so that I'm not able to answer that question. Okay. So last thing I'm going to mention are VX152 and VX40. I'm going to try and move fast because I'm almost out of time. These are other corrector molecules that may help improve the effectiveness, once again, of either a VX661 or potentially Orkambi. In preclinical trials, triple combo therapy, where you took a 3-drug cocktail as opposed to a 2-drug cocktail worked about 3 times better than Orkambi in terms of chloride flux. And so this would be a change in sweat chloride of about 30, which once again may be more significant. So once again, so here's triple therapy and here's Kalydeco. Suddenly we're not just doing 10, we're doing 30. We're starting to see bigger effects clinically, and at least that's the hope. You know, when we start to deal with drugs that do a better job of correcting, we'll get better changes in sweat chloride. We don't know. There are phase 2 trials planned later this year of these drugs. We will see whether they work well in phase 2 trials, whether they have side effects, et cetera, et cetera, et cetera. It's too early to know, but that stuff is coming down the pike and are actively being planned. So my conclusion, the second generation corrector VX661 is in phase 3 trials. We're hoping it's going to improve slightly on Lumacaftor. The third generation of triple therapy may dramatically improve F508 del rescue. There are other things like GSNOR drugs, which may boost Orkambi in other ways. So there's a lot of exciting stuff coming down the pike in the next year, 2 years, that we're actively studying right now. And once again, this plug that when you're able, we love getting people involved in research, because this is how we move the thing forward, and we couldn't move this field forward without the patients who are willing to do research and help us figure out whether these things work or not. And I think that is pretty much what I had.
Awesome. Thank you, Ted. Do you want to introduce your colleague?
I will introduce my colleague. So I'd like to introduce Dr. Zuckerman, who is the senior adult CF doctor. I'm the junior adult CF doctor. He's been here for 15 years, and he's the director of the adult program at Maine Medical Center, and my mentor, Dr. Jonathan Zuckerman.
All right, well, I'm going to be going on a little bit of a meandering talk today. I've actually been ruminating quite a bit about how we cover. There's so much going on in CF right now in terms of basic research, and I think Ted did a great job of covering the next few steps from the development of Orkambi for molecular therapies for CF. But some people in the audience may be wondering, about what's happened to gene therapy. Is there any role for stem cell biology? And then something that I've become increasingly interested in the care of CF patients is personalized care. So we're actually going to go away from science a little bit towards the end of the talk, and I'm hoping that that's successful. All right, let's see. Okay, so disclosures. I have been working on some projects with Vertex and Anthera, and I'm on the scientific advisory board for Gilead, but full disclosure is that I'm hoping that you're going to grow in your interest with respect to our new approaches for CF care. So this is one of my favorite slides. Many years ago, we had Beth Suffian come to speak to us by webcast. How many people in the room know who Beth Suffian is? That's great. So she's pretty famous. She's a person with CF who's a lawyer. She works for the foundation and helps a lot of adults navigate through the difficulties of getting disability, having their insurance issues straightened out. And what she showed us is that when she was a kid, her treatment for CF was up in the upper left-hand corner. It's just a cup of tea, lemon, and honey. And we've come so far since that time. Now we have all sorts of therapies that we've tried to standardize in some respect over here. And then, as Emily eloquently described, the blue lightning pill that we have now down at the bottom. I think for most patients with CF, they envision a future where maybe you only need to take pills and not do any inhaler therapies, or perhaps have a one-time cure. So that's always something that's on our radar screen. So right now, why do we standardize care? This is an issue that doesn't just affect CF patients. It really cuts across medicine in the United States overall. But it allows us ways to improve the care of patients by raising at least the general standard of what's provided to people. It improves efficiency. It gives the healthcare team a convenient framework around which to think about care, and it may make our care more uniform across healthcare systems. So along the lines of standardized care, we also have guidelines, and the CF Foundation has been very proactive in developing guidelines. Can everybody hear me okay? No.
Stay near it.
Stay near the mic. The guidelines have helped us with respect to respiratory care, nutrition, exercise, and social work. The problem is, and how many people have read about the sneeches? Something that is in style at one point may fall out of style at another point. So sometimes what we have that happens with standardized care, it's our best approximation of what should be done, but then 5 years later we find that maybe we were a little bit off target. And how many people have heard about vitamin D? It's in, it's out, cholesterol in and out. So we have to be aware that just because you're getting standardized care doesn't necessarily mean you're getting the best care. Our goal in implementing care in a standardized way is that it should be intentional, not just an assembly line. And we're going to get back to that a little bit more later. Evidence-based medicine is a conscientious, explicit, and judicious use of current best evidence in making decisions about the care of individual patients. And that's a really tricky thing for healthcare teams to do, to think about the individual. Sometimes things have to be custom fit. Now, last year I was very excited. I gave grand rounds at Maine Medical Center, and I think it was the day before my talk, Barack Obama gave a State of the Union address, and he had actually a CF patient stand up. Does anybody remember that talk? Yeah, I thought that this was brilliant. I was very excited to hear him talk about a term he called precision medicine. And in the final sentence, he said, in some patients with cystic fibrosis, this approach has reversed a disease once thought unstoppable. And I thought a little bit about the notion of precision medicine. I actually like the term personalized medicine a lot more. And we're going to touch on that. Dr. Sears was really touching on precision medicine that's largely grounded in the study of genetic mutations. And so we're trying to customize treatments to people who have various genetic predispositions to disease. Personalized medicine is different. And just think about that while I go through the rest of the talk. I'm going to come back and tell you what I think personalized medicine is a little bit later. But before we do that, I'm actually going to launch us into some what seems like science fiction here for a little bit. We're going to talk about gene therapy and stem cell biology for a little bit. Okay, so the story begins back in 1989. This is when I was still doing my medical residency. I didn't even really think much about CF at that time. But the gene was cloned in 1989, and the abnormality is found on the long arm of chromosome 7, and the protein that Dr. Sears was talking about, the CFTR protein, is a protein that we for many years have thought about as inserting into the lining cells of many of the organs in our body. So the airway lining cells, the lining cells in our GI tract, the reproductive tract. I'm going to give you some surprises in a few minutes, but for the most part, we have thought of the CFTR protein residing in the tippity top of these airway lining cells. That's where the major problem is. And these beautiful green projections are the cilia, which move mucus up the airways. So we think that CFTR is mainly doing its work along this red stripe that's called the terminal bar. Now, the three major labs that were involved in the discovery of this gene actually crossed boundaries between the United States and Canada. Dr. Francis Collins, who's become quite a bit of a rock star himself at the CF Foundation, is now the head of NIH, and he was the lead scientist on the Human Genome Project. For those of you who've heard about the Human Genome Project, he was part of the governmental research team for that. He was a co-discoverer of the CF gene, and I see him as somewhat of a prophet, seeing where the field of medicine is going in CF. Now, shortly after this team discovered the CF gene, I was working at the University of Pennsylvania, and we were working on gene therapy. Everybody at that time thought that gene therapy was going to be the cure for CF, and the head of our lab had a very simplistic view of how to fix the problem. He said, I don't really care what CFTR does. We know that this is a recessive disease. What that means is if you just correct one copy of the gene in the cells, you've corrected the problem. So you don't have to understand the biology. If you could just get that blasted gene in, it should work. And actually in the test tube, it works great. We had no problems getting transfecting cells. At that time, we were using adenovirus. And we would cure the CF problem in these test tube cells. The problem is our body is designed to reject foreign invaders, foreign DNA. That's why we fight viruses, viral infections. You may fool me once, right? But after I'm vaccinated or infected, I don't get infected with the same virus again. So we actually had to take a big step back. This is back in the early 1990s, just to give you a little bit of a time frame. Almost 20 years ago, actually it was the mid-1990s, that we found that gene therapy was not working the way we had designed it. We had to go back to the drawing board and understand what's actually going on in the cells. And I've actually updated this slide since I last showed it at Maine Medical Center. What I had down here was we had to learn more about airway biology. And now I'm realizing we have to learn more about cell biology in general. Because as it turns out, we just covered this in our journal club a week or so ago, it looks like CFTR is not only expressed in the airway lining cells, it's actually also expressed in the muscle cells that line the airway. And so people with asthma, they get bronchospasm because the muscles contract. We now know that CFTR is expressed in the muscle cells of the airway. And that may be one of the reasons why when we give a medicine like Kalydeco, there's an almost immediate response, much faster than you'd expect if somebody just had lots of mucus and airway remodeling with scar tissue. There's actually some bronchospasm. So that I thought was a fascinating thing. We're learning more about the biology of this disease every year. Now, how much CFTR function is enough? I'm going to blow through this slide quickly because Dr. Sears kind of covered it nicely in his talk. But basically, we realize that we don't have to correct all of the CFTR dysfunction. Somebody who has about half of the wild type amount of CFTR function, for all intents and purposes, looks normal. So our strategies don't have to get you up to 100%, but we think that in patients with G551D, we're getting about 50% correction, and we probably don't even need that much for people to really notice a difference with other mutations. Now, one of the things that I thought was so inspiring at the CF Foundation, I think it would be pretty easy to say, you know what, we've gotten a couple of good products here. We're going to focus on the people who have the mutations that are most frequent. You know, F508del, we want to do a lot of work on that. The problem is there are about 2,000 mutations that have been identified that cause CF, and some of them are very, very rare. And some of them are so rare that you'd never be able to do scientifically adequate clinical trials with placebo and drug in enough patients to get valid results. So you could just hang up your shoes and say, you know what, we'll just do standard therapy for those people that we talked about. Standard therapy is pretty effective, but the CF Foundation actually is more visionary than that. And I'm very impressed that they feel, as part of one of their mission statements, they want to try to find a treatment for every patient with CF. And some of these may be tough nuts to crack, but they're not giving up on it. And one of the ways, and I'm going to actually, in the interest of time, go through these slides very quickly. We should be able to have these things up on the web at some point so that people can look them over. But if you notice these different colored circles, these are trying to indicate graphically the percentage of patients with different types of mutations. So there are different strategies being taken for different types of mutations. And notice as we get to the end, the whole grid is filled with different strategies to treat different mutations. And so that's all the more reason for all of the patients, well, we don't have too many patients in here, but families and patients to know what the gene